constitutive sort of behavioral traits that sort of drive the variation we see. And the reason that I think that this is such an interesting behavior is that aggregative behavior can be seen across the animal kingdom, and not only can pretty much any taxa you look at will have one or more representative species that aggregate, but there's a bunch of different hypotheses and reasons for why it's adapted. So you have the predator defense that you get from the improved aposematic signal, these firebugs. You have the better mating efficiency that lekking birds receive. You have the improved ability to manage microenvironment, like in these bed bugs. And you have the improved foraging efficiency through pack hunting, like in wild dogs. And what's really cool is, in some cases, like these pink caterpillars, you'll have an argument to be made that multiple of these factors are playing a role in the additiveness of activation. But one of the things that's sort of left open is if one or more of these is a more common driving factor in the evolution of aggregatedness, or if one sort of is the key driver. And to address these sorts of questions, what would be helpful is a group of closely related species that sort of vary in their aggregative tendency. And a great group to study those in is pine sawflies in the genus Neodiprion. So they vary in a bunch of interesting traits, like the time of diapause, the color and pattern of the larvae, and for the talk today, the most important factors that they vary in is the ovipositioning pattern of the female. So not only how many eggs she's laying inside of a needle, but also is she laying them on a single branch or sort of spreading them across a tree or multiple trees, and also the variation in the aggregative behavior of the larvae. And an important thing to note is that these are minor forestry pests, or at least some species are. And so there's a lot of historic work describing these variations and actually most of the variation here. So there's a lot of historic data that can sort of be looked at. And so the historic classifications of the social behavior of these larvae has mostly been categorical, with a lot of species, as you can see, sort of being described as gregarious. And what you'll see when species is described as gregarious is they'll be forming these large feeding aggregations. Very noticeable. There's a lot in a single place. And then fewer, but a not insignificant number, are categorized as intermediate or solitary, where they're forming these smaller aggregations or occurring singly on the branch. But the issue here is that gregariousness is, calling something gregarious or solitary is sort of implying something about the behavior of the larvae. But what these descriptions actually seem to be describing is the colony size. And the reason that's important is that colony size can be thought of as sort of a composite trait of both larval behaviors, like aggregated tendency, or whether or not the larvae have a tendency to group together closely or sort of spread out, as well as the egg clumping behavior of the mother, or whether she's laying all of her eggs in very close proximity to each other, or sort of spreading them out across a tree or multiple trees. And you can kind of imagine that the trait of a large colony could be driven by some sort of combination of these two traits working together. And so what I'm hoping to sort of demonstrate today is that these historic categories are describing specifically colony size, and even then they're still leaving a lot of variations that have been described, and also sort of try to investigate which of the other two traits are sort of driving the variation we see in colony size. So to do that, I collected data from a monophyletic clade of about 19 species that occur in eastern North America, and these three traits. And we're going to start with colony size, which we sort of define as the count of individual larvae found in a group sort of on the same branch. And this was assembled through field observations as well as the literature records. And if we plot those values out on the graph, you can see that across the species, there's sort of continuous variation. So on the left here are colony sizes that are quite small, and on the right are colony sizes that are much larger. And you can see that to historic categories, gregarious is sort of matching this well, but it's still leaving all of this variation sort of left without a description. The next thing I'd like to bring up is the larval aggregative tendency. And so to measure that, we put 5 larvae in a petri dish and recorded them for 90 minutes. This sort of gives you an idea of what that looks like. Here's a time-lapse sort of GIF of those videos. And so over the course of those 90 minutes, we recorded the distance between the head caps of all the larvae. And even from these sort of simple videos, you can see top two are aggregating quite quickly and sort of remaining close together, while these bottom two are sort of wandering around with this lot less interest in sort of grouping together. And then when we plot out those singular values we get, so we average all those pairwise distances together across the video, and then average all the figures for species. Again, we see a nice continuous distribution, but we see that the historic categories aren't actually describing the aggregative tendency of the larvae very well. And then the final trait to the egg clumping, which is sort of the count of eggs in a cluster. So sort of like the colony size, it's the number of eggs that we find on a single branch sort of close together. And that's from field observations and literature again, but also from a few positioning experiments. So here it is plotted out again. Very similar conclusions to the colony size, which kind of leads us to believe that these two may be highly related. So continuous variation in the historic categories are sort of accurate, but leaving a lot of undescribed variation. To give you an idea of how complete our data was, we do have data points for all three traits and all species except for 3 and 8 clumping. But then to actually start to address how related these are, we did linear regression. We did that both with and without phylogenetic correction. And so first we're going to look at without phylogenetic correction. And then when we look at aggregated tendency and colony size, we find very little evidence, pretty much no evidence of any sort of relationship between those two traits. And this is in contrast with the egg clumping and colony size, where we find a very strong relationship between those two. However, these are still very closely related species, so we should also be looking at this in a phylogenetic context. And to do that, we compared, we first searched for a phylogenetic signal by comparing models of evolution of the traits where each species is considered to be independently evolving versus one from sister species and closely related taxa to one where the closely related species are presumed to have shared evolutionary history. And from this, for colony size, we do find strong evidence for phylogenetic signal, and that's sort of exemplified by the fact that, you have like this clade up here where they're all forming pretty small colonies, and this one down here where, on average, they're forming much larger sized colonies. And then, but for aggregated tendency, we find pretty much no evidence of a phylogenetic signal, and you can kind of get an idea of that by the fact that the least aggregated species are sort of spread out across the tree, as are the most aggregated species. And then the final trait is egg clumping, and we find possible evidence for a phylogenetic signal. So it's not significant, but it does seem to be trending in that direction. And the fact that we don't find a significant evidence for phylogenetic signal could be related to the fact that we are missing 3 data points here, or possibly a few inaccuracies in the specific data points we're using. But even still, we see things like this clade with low numbers of eggs clumped together in this clade with much larger eggs. And then finally, what we looked at is looking for correlated evolution between these. So we're comparing a model where colony size is evolving as an independent trait to one where it has correlated evolution with these other traits, and find that the model isn't improved by including aggregative tendency. The model is significantly improved by including egg clumping as a correlated trait. And if we include both together, we find that it doesn't significantly improve it over the egg clumping model or not. And so from this, we can get a few conclusions. So historic categories of gregarious do seem to be reflecting colony size, but there's still a lot of variation that's left undescribed, because colony size and aggregative tendency and egg clumping are all sort of having this continuous variation across the genus. And also, colony size and possibly egg clumping do have some phylogenetic signal, and the variation in colony sizes seems to primarily be driven by egg clumping behavior. And the reason that's important is it leads to the next question of what's driving the selection on group size. So the females aren't necessarily long-lived on average across the genus. It's maybe a week at best that you can expect them to be alive. And so there may be some sort of evolutionary selection going on to have them sort of, if they find a suitable evolutionary site, to lay as many eggs there as possible rather than risk failing to find another site, or have risking death before they find another site. And so that could even be selecting for large group sizes in a way, even if there isn't benefit for larvae. But the larvae are still grouping together, at least in some species. And so that leads to the question of, is there some sort of, there's presumably some sort of benefit to larvae of aggregated behavior. And so the future directions that what we do is to look for some sort of relationship between those three traits, especially larval aggregative tendency, since that's most directly measuring the larvae's preference for each other, and looking at how it correlates with some of those other variable traits like color to get an idea of the possible benefits such as coordinated defense and signaling, host use, and maintenance of optimal microenvironment, as well as the possible cost to that group living incurs, such as increased disease risk. And so I'd like to acknowledge my funding sources